Hello everyone. Today we are going to be talking. Hello, sorry. Today we're going to be talking about enzymes. Enzymes are one of the most important topics within this website, as they allow us to understand more complex topics and concepts. See, enzymes, as we were talking about macromolecules before, enzymes are a form of protein. They are a form of protein. They catalyze reactions. They break bonds apart. They break things. For example, in carbohydrates, they break glycosidic bonds apart. They can even rejoin them. These are through the process of catabolic and anabolic reactions, which we're going to discuss. However, let's actually look at what enzymes do. So enzyme is a form of protein. Okay, enzymes are a form of protein. Enzymes come in all shapes and sizes. However, this is my example of it. So you see this area right here? Let's color this right here. This area right here that we just outlined right now. This area, I use a different marker here. It's called the active site. This is called the active site. And another, let me use a different marker actually. And what is the point of the active site? The main point of the active site is to attach substrates within that active site and catalyze reactions. We'll talk about inhibitors and cofactors and coenzymes in a couple of minutes. However, this is the basic. So basically, substrates will attach themselves to active sites, and a substrate has to be exactly, or the precise exact substrate to be within that active site. And that active site has to be broken apart. No, wait, sorry, my apologies. I meant to say, when that substrate, sorry, let's draw this out. Sorry, I tried replicating the same enzyme. Anyways, when that substrate and the enzyme bind together, there's things called products. Products can be made or they can be broken apart. That depends on what type of reaction or what type of catalyzation the enzyme wants. And yeah, products are made at the end. That is the fundamentals of every enzyme. And yeah, that's basically the fundamentals of enzymes. So there's something also called an induced fit. What is an induced fit? So basically, like we were talking about earlier, that's the substrate. When this substrate attaches to this enzyme, the enzyme will undergo some changes due to thermodynamic rules. This enzyme right here, it will undergo some changes to make the substrate fit almost perfectly to the enzyme. This is called induced fit, and this allows the active site and the substrate to bind more properly together. Because even though there are bonds like hydrogen bonds and other form of bonds, this induced fit allows the substrate and enzyme to perform the catalyzation much better, or the process of breaking down substances or breaking down the substrate or forming the substrate much better. It allows the enzyme to do that job much better. So there's a lot of factors that could affect the function of enzymes. What is that? Firstly, the pH. All right, the pH affects how enzymes function. The temperature, the external or internal temperature, basically shows what functions the enzymes are open to. Because if the temperature is too cold or too hot, enzymes cannot function. However, the ideal temperature for human enzymes is 37 degrees. Another one that is also a really big factor is if there are a lot of enzymes within an area, There are a lot of enzymes. There are a lot of enzymes within an environment. They can catalyze a lot of reactions too. And if there are less, then they cannot catalyze as many reactions. So the main point of enzymes is they lower the activation energy for most reactions. The lower the activation energy is, the faster the enzymes speed up the reactions, because due to low energy, enzymes can speed up reactions by by a significant amount actually. Next, let's look at how enzymes actually work within macromolecules. So for example, let's look at lactase. All right. Sorry about my drawings. These are just two simple monosaccharides. Anyways, this right here is a lactose. This is a disaccharide like we discussed earlier. This is a disaccharide that has two monosaccharides connected with a glycosidic link. So what this enzyme is going to do, this enzyme is called lactase. So you see, our bodies cannot digest lactose as efficiently without lactase, because lactase has to break this disaccharide down, so our bodies can use it for many processes such as cellular respiration and other biological processes. Lactase breaks lactose down into two monosaccharides. Let's just name them. The first one is galactose and the second one is glucose. These two are monosaccharides that are bonded with a glycosidic link, and that makes lactose. So you see, many people do not have the enzyme called lactase. That's why they are lactose intolerant. That's why their bodies cannot process lactose, as they are a big carbohydrate, or a big, not a big carbohydrate. Sorry, they're a disaccharide that is very hard to use for energy. So lactase, it breaks this link apart and allows this glycosidic lactase breaks this glycosidic link apart and puts it into two different monosaccharides that our body can digest. Okay, all right. There are many different types of other enzymes. For example, within lipids, there's lipase. Within starches and carbohydrates, there's amylase. It's within our mouth. And for proteins, there's enzymes called protease. Anyways, there's other things that also affect enzymes' function. They're called coenzymes and cofactors. Let's write that down. Cofactors. Cofactors are factors. Cofactors are factors such as metal ions. Sorry about that. Metal ions. It can be iron and coenzymes. This can be organic molecules. Organic molecules such as vitamins. Vitamins are a great source of, a great example of coenzymes. So when these factors as these factors bind to the substrate or active site, they help with the process of creating or breaking down substrates into products, like we mentioned earlier. So now that we have a sort of brief overview of what cofactors and coenzymes are, let's put them to use. Let's look at how cofactors and coenzymes work. So, firstly, coenzymes. Let's draw this out. For example, if this is the enzyme, Oh my goodness, that's a horrible enzyme actually. My apologies. I don't know what I'm doing. Okay, much better. All right. And suppose this wants to fit here. However, if a cofactor or what cofactors and coenzymes essentially do is they help. One second. For example, what if it looked the substrate? This is the substrate right here. Let's just color this. This is the substrate right here. This is the cofactor, and this is the enzyme. Coenzymes and cofactors, basically what they do is they help the substrate bind better to the enzyme so the enzyme can perform its job better. So along with induced fit, coenzymes and cofactors also help. However, what happens when cofactors and coenzymes do not want substrates to bind to the enzyme? These are called inhibitors. So there's two types of inhibitors. Firstly, there's things called competitive inhibitors. And there's things called non-competitive. And non-competitive inhibitors. So basically, there's two types of inhibitors. So what competitive inhibitors do is within, let's draw this out again. For example, this is an enzyme. This is an enzyme. This is the active site right here, okay? This is the active site. What competitive inhibitors do is they are found within the active site, and they do not allow their job, like they do not allow substrates to bind within that active site to perform their functions. This can be for good reasons or bad reasons, which we'll get onto in later processes. And non-competitive inhibitors. So this was for the first one, for the bottom one right here. Non-competitive inhibitors. This is the active site. Once again, this is the active site. Sorry for my enzyme drawings. I'm pretty bad at them. This right here, for example, if a non-competitive inhibitors bind to other areas of the enzyme, these areas that are not the active site will be called allosteric sites. If these non-competitive inhibitors are attached to these allosteric sites, they can denature the enzyme, and they can cause its change. It can cause the enzyme to change in many ways. This can allow the substrate, due to this, substrates, for example, if this non-competitive inhibitor gets attached to this enzyme, it denatures the enzyme, which is really bad, and this substrate cannot join or connect with that active site anymore. It can happen for a good reason again, or a bad reason. However, if the substrate still attaches to the active site even after the non-competitive inhibitor has denatured the enzyme, that means the enzyme may be bad itself, or may produce bad results within biological compounds, molecules, or any other biological process that it carries out. So it's very important to understand this, and that's basically really important to understand, as we will get on to the final topic of enzymes, which is called feedback inhibition. For example, if enzyme 1 converts substrate A, so let's see, this is the active site right here. This is This is the substrate, and this is the enzyme. Same thing here. Enzyme A, let's say B and C. So first thing is when enzyme A converts A to intermediate B, it converts the substrate from A to B. It creates it helps with the process of making products. So let's just write this here. Slowly starts. And when B goes to C, which is the final product, and by the way, I forgot to mention, I think, products can be broken apart. Products can be broken apart, or they can be formed, which is catabolic and anabolic reactions. When they're broken apart, they're called catabolic reactions. When they're brought again, or when they are brought together for many bonds to occur, this can also be referred to as an anabolic reaction. Anyways, back to the main part. When intermediate C has taken place, many things can happen. So firstly, if too much of product C is formed, your body may not like that, and it's harmful to your body. So product C can become a non-competitive inhibitor for enzyme A. When it becomes a non-competitive inhibitor, what did we talk about earlier? So this product right here can join can bind to the allosteric sites here, or it can join to the allosteric sites right here, and it can basically denature this whole enzyme. As the product from C denatures the whole enzyme, the substrate can no longer bind with that particular enzyme, which stops this whole chain of intermediate reactions. So basically, that's how if our body has too many products, that's how it stops it. Thank you for watching this video. Hopefully it helped you regarding confusions and other information regarding enzymes. Thank you so much for sorry about that. Thank you so much for watching. Have a nice day, everyone. We will continue more complex topics in the future. Have a nice day.